So hey students, today we are going to do some MCQs from Biotechnology Principle and Processes. Okay, so our first question is rising of dough is due to production of CO2. An enzyme catalyzing the removal of nucleotide from the DNA is exonuclease. From the ends. Okay. An enzyme catalyzing the removal of nucleotides from the end of the DNA is exonuclease. The transfer of genetic material from one bacterium to another through the mediation of vectors like virus is termed as transduction. Which one of the given statement is correct in the context of observing DNA separated by agarose gel electrophoresis? So our answer is D. Erythium bromide stained DNA can be seen under exposure to UV light. Restriction in restrictive enzyme refers to prevention of the multiplication of bacteriophage in bacteria. Restriction. A recombinant DNA molecule can be produced in the absence of following. In the absence of E. coli, you can do and make a recombinant DNA molecule. Okay. In agarose gel electrophoresis, DNA molecules are separated on the basis of their size only. Smaller and larger size. Okay. The smaller the size, faster it moves. The most important feature in plasmid to be used as a vector is origin of replication, ORI. While isolating DNA from bacteria, which of the following enzyme is not used? So its deoxyribonucleases is not used. And which are used during DNA isolating DNA from bacteria is lysozyme, ribonuclease and proteases are used, but it is not used. So which one is not used? So D is not used. Which one of the following has popularized the PCR polymerase chain reaction? Availability of thermostable DNA polymerase, Taq polymerase. An antibiotic resistance gene in vector usually helps in selection of transformed cells. Significance of heat shock method in bacterial transformation is to facilitate uptake of DNA through transient pores in the bacterial cell wall. The role of DNA ligase in the construction of the recombinant DNA molecule is formation of phosphodiester bond between two DNA fragments. Which one of the following bacteria is not a source of restrictive endonuclease? So C is not. Okay. But you can use Haemophilus influenza, Escherichia coli and Bacterius amyloili, source of restrictive endonuclease, and not is this one. Which of the following steps are catalyzed by Taq-Pol in PCR reaction? So our answer is extension of the primer at the end on the template DNA. You can learn the PCR reaction by denaturation and annealing and extension, DAE. A bacterial cell was transformed within a recombinant DNA that was generated using human gene. However, the transformed cell did not produce the desired protein. Reasons could be A. Human gene may have introns which bacteria cannot process. As we know that human genes does not have introns as they are removed during splicing. Which of the following should be chosen for best yield if one were to produce a recombinant protein in large amounts? So our answer goes to a continuous culture system. Who among the following was the awarded the Nobel Prize for development of PCR technique? So our answer is Carrie Mullis was awarded for developing PCR technique. Which of the following statement does not hold true for restrictive enzyme? That is C. It is isolated from viruses. It is not isolated from viruses. Okay, thank you. All the best for exams.